Hi, everybody. Welcome back. We're still doing gene regulation. We just covered prokaryotes. Now we have to cover eukaryotes. How do they regulate their genes? We don't have circular DNA. We have stranded DNA. We have 46 chromosomes. And each of those needs to be highly regulated. You don't want arm genes turning on in legs, and you don't want liver genes turning on in the pancreas, although there is a little bit of overlap, as we discussed earlier, as to what they would have on, like membrane and enzymes. So we're going to divide eukaryotes up into three different categories for regulation. There's going to be pre-transcription, post-transcription, and then post-translation. All three steps come with their own little gadgets on how to regulate which gene's being expressed, how much of it's being expressed, and what do you do with the products? Do the products come back and inhibit? Do the products build up? Do they diminish? What happens? So we're going to start with first the pre-transcription. Your DNA, if this is a strand of DNA without putting a helix on it, really is wound together with these little guys called histones. Histones are special proteins that are set up in groups of 8. So there's 1, 2, 3, 4, and then there's 5, 6 back there, 7, and 8. And together these make like a little thing called a nucleosome. Well, that's how the DNA gets wound up. So you got the DNA winding this way, and then it goes around, and then the DNA goes back out this way. That's just to keep it organized. Well, you can methylate those histones, meaning you can attach little methyl groups, and the methyl groups will attach to the C's of DNA. So if this is the strand of DNA, a methylated strand of DNA would look like this. It would get really wound up tight. Methylated, and it becomes something called heterochromatin. Heterochromatin is like gluing chapters shut. I have your textbook right here, and if I were going to be a master at genetics, or if I was the genetic part of the body, almost there. Here's the beginning of the genetics unit, right? And I would leave the genetics portion open. Wow, I really nailed that. All the way down to right about there. So this is the stuff I'd want to keep open. I would glue all of this shut so I couldn't have access to it. I'd glue the rest of it shut so I couldn't have access to it, but I could have access to this, right? That's no different than what I'm describing. The parts that you can't use, or let's say we're inside a kidney cell, we're going to heterochromatin the snot. I said kidney, right? We're going to heterochromatin the snot out of the eyeball genes and the liver genes and the stuff that doesn't belong genes. But we're going to loosen up and allow access to what we call acetylate. Those histones on the regions we do want read. It loosens it up, it opens it up, and so it can be read. So this can be read, and this cannot. The acetylation loosens up the chromatins, it loosens up the histones from each other so that now it can be read. The methylation condenses it down. It compacts it so that the DNA cannot be read. This is a pre-transcription. This is a good word to know right now, epigenetic. Epigenetic means we are affecting expression without changing the DNA. Meaning we're not turning on or getting rid of or substituting ATCs or Gs. We're just deciding which ones can be read. That's an epigenetic. We need to know that phrase for this chapter. That's an epigenetic way of controlling gene expression. Okay. The next one are using something called enhancers. Enhancers, if we go back to transcription, there was a video where the mRNA was resting and you had a pile of transcription factors, and then the woman narrating it said 3, 2, 1. And what you saw was a stretch of DNA come down and touch those transcription factors and cause the mRNA to be read. What they were showing were enhancers. Enhancers are regions upstream that will gather little activators, and they bend down to activate the gene. Let me show you what I mean. Here's the enhancers, and the enhancers have an affinity for very specific activators. Those activators, once they aggregate at the enhancer site, will bend down and make the mediator proteins and transcription factors fit better at the promoter site so that the gene can be read. Without the enhancers, the gene doesn't get read as much. Maybe I'm not going to give you a percentage, but it does not get read as well, as much. The enhancers do just that. They enhance the reading of the gene. Now RNA polymerase can come in and it will read and it can transcribe the gene. Enhancers are different for different tissues that we have. So on the left-hand side, we have a liver cell, and on the right-hand side, we have the lens cell of your eye. In the liver cell, we have three types of enhancers. In the lens cell, we too have three types, and there's one that crosses over, right? But in the liver cell, it has genes that are turned on to make these specific activators to enhance the reading of liver genes. In the lens cell, they have their own little specific pile of activators to enhance only the reading of lens genes. This is a way to keep your hand from staying in your hand and your foot stay in your foot. Those genes are only turned on in certain tissues. So that's all pre-transcription. Heterochromatin, euchromatin as it's called when it can be read. I should go back and write that in because you probably were going to ask about that. Let me go back. So if this was heterochromatin, this stuff that can be read is called euchromatin, as in true chromatin, eukaryotes. Post-transcription, what happens after the mRNA reads? Well, we've already done some of that. This is where the introns and the exons go. You can put a poly A tail on there, and that'll affect how long the gene is read for. Everything needs a poly cap, a 5 cap, so that it does not get degraded. But we also talked about alternative splicing a little bit. Here's something called troponin. Troponin is inside your muscle cells, and it helps with muscle contraction. Troponin T is the gene right there. Some of the troponin gene, when you include 1, 2, 3, and 5 exons, this will become the actual troponin T, what you see over here. But when you get rid of 3 and we use 4 instead, this becomes a different type of troponin that is used in embryonic tissue and won't be used again later. So this stuff gets used early on. This stuff gets used as adulthood. This is how bacteria have 4,000 genes and we only have 22,000. We're not 5 times more complicated. We're far more complicated than bacteria. We have hundreds of thousands of capabilities inside our gene because we can alternatively splice. One gene can make up to 6, 4, sometimes 7 different types of proteins just through alternative splicing. So let's write it in. Alternative. Let me do a better job. I'm writing kind of sloppy today. Don't know what it is. Changing the linear sequence of exons. And I write it that way As I've said before, you can do 1, 2, 3, 5, but you can't do 1, 3, 5, 3. You can't reverse them. You can only go in the linear sequence. Okay, so that was post-transcription. Poly A cap, poly A tail, 5 cap. 5 cap has to be there. That shouldn't count. So the poly A tail and alternative splicing. And now we get into post-translation. You've made the protein. What now? Well, you can make these little transcripts of RNA, and these are called microRNAs. They're not going to use mRNA because obviously it's already been taken. But these miRNAs, they get transcribed and they fold back on themselves. Right? So there's base pairing that's involved. And then a special enzyme called dicer comes along and it cuts it. So we're left with one side and the other side. The way that this can work is post-transcription, pre-transcription, microRNA can slide into the bottom of the ribosome, right? So you have the bottom and you have the top. This little guy can get inside that groove and prevent any more translation from occurring. That translation, now you can see this is the mRNA, there's my poly A tail, doesn't go because it binds up with this little guy and it almost acts like anticodons and seizes up the mRNA from being read anymore. After these proteins are made, this is a protein that you want to make yourself familiar with. Ubiquitin comes from the word ubiquitous. When things are ubiquitous, they are everywhere at all times. They're just everywhere. So ubiquitin is the protein that is everywhere at all times. The way it works is it attaches itself to old or excessive proteins, and then there is a proteasome, a little digestive protein, that will break that dude down, digest it. That is a way to regulate the amount of protein that is available inside the cell, the amount of product. All right, go in the opposite direction. There's these little proteins called chaperonins. I think I'm getting the spelling correctly. And we learned those when we did chapter 4 for polypeptides. You have your secondary, maybe getting close to your tertiary, that guy goes in there, and this creates the proper microclimate, if you will, for the hydrophobic interactions to happen, for the hydrophilic tertiary interactions to happen. And when it comes out, it is in its final configuration. So this is a post-translation modification that can be made to proteins to further change or regulate what type of protein is being made. Again, these are divided up into threes. Pre-transcription, what happens after transcription, and then what happens after the protein is made. I think if it were up to me, I would put the miRNA somewhere between transcription and translation, because it does slide in there. It's put in with translation because that's right where translation's happening. So it's the moment of translation, whatever. I'll accept either one. Here's a nice way to look at it. Pre-transcription, methylation, heterochromatin, euchromatin, transcription factors themselves, whether they're available for the promoter region to enhance RNA polymerase from coming over and reading or not, right? The transcription factors aren't there, the gene isn't getting read. And enhancers, those were the upstream things that will bend down with their activators and activate gene production. Post-transcription, you got to have your poly A tails. The length is going to be the lifespan of how long that guy gets read, or girl, gender neutral. There's alternative splicing in linear fashion. You can go 1, 2, 3. You can go 1, 3. You can go 2, 3. But you can't go 3, 2. That is a no-no. You can't go backward. Post-translation, microRNAs that get inside the ribosome and block further translation. Ubiquitin are proteins that mark excess proteins for death. And then RNA nucleases are rattling around everywhere. They're ready to break down free RNAs that don't have a polycap. So once an RNA shows up without that 5 cap, these These RNA nucleases, their job is just to start chopping and whopping and cutting up that RNA because we don't want information that doesn't belong just laying around. It might get read and do something wrong. Cool? I think so. Okay, so that sums up our gene regulation for both prokaryotes and eukaryotes. I'm going to throw up a couple little extra videos because I know you guys like to get a little extra on the side for information, but this is the bulk of the chapter. There's a second half. It has to do with development. I want to save that for the next week. This is our focus this week, getting down the types of regulation for prokaryotes, operons and introns, and for eukaryotes, the ones we've just described today. All right, I'll be talking to you guys soon. Have a great day. Bye-bye.